Hello student, today I am going to teach you on the topic immunohematology. Introduction. Immunohematology is a division of hematology concerned with the study of immunological or antigen-antibody reaction and their effect on blood. Blood is a body fluid found in humans and other animals that carries oxygen and nutrients to the tissues and organs. It transports metabolic waste product away from your body tissue. It is made up of several main components including red blood cells, white blood cells, platelet, and plasma. Blood loss may occur due to injury, major surgery, or diseases that destroy red blood cells or platelets. So in order to compensate blood loss, blood transfusion plays an important role for our survival. Experiments with blood transfusion have been carried out for hundreds of years. Many patients have died, and it was not until 1900 when Austrian scientist Karl Landsteiner discovered human blood groups that blood transfusion became safer. Blood group antigen. Blood is classified into different groups according to the presence or absence of molecules called antigens on the surface of red blood cells in a person's body. Antigens determine blood type and can either be proteins or complexes of sugar molecules or polysaccharide. Types of blood groups. There are 35 types of genetically determined blood group systems. Among them, the ABO and rhesus, that is RH systems, are the most important ones used for blood transfusions. The ABO blood group system were first identified by Austrian immunologist Karl Landsteiner in 1900. ABO system. There are four main blood groups defined by the ABO system. Blood group A has A antigens on the red blood cells with anti-B antibodies in the plasma. Blood group B has B antigens with anti-A antibodies in the plasma. Blood group AB has both A and B antigen but no antibodies in the plasma. Blood group O has no antigens but both anti-A and anti-B antibodies in the plasma. Group AB was discovered in 1902 by Landsteiner pupils Alfred von de Castello and Adriano Sterli. ABO antigen is present on the surface of red cells as well as tissue and endothelial cells in the body. It is also found in soluble form in plasma and other body secretions in people known as secretors. 85% of the populations are secretors, so non-secretors are in a minority. RH system. It is discovered in 1940 by Landsteiner and Wiener. It is most complex erythrocyte antigen system. RH gene is located on chromosome 1 and found exclusively on surface of RBC. Landsteiner and Wiener saw that antibodies produced against the rhesus monkey red blood cells agglutinated the red blood cells of 85% of a human population. The person who have D antigens are said to be RH positive, and the person who doesn't have D antigens are called RH negative. It lacks corresponding naturally occurring antibodies in serum. It is produced upon sensitization and is of immunoglobulin G or IgG isotype, which can cross the placenta. Antigen. Any substance foreign to the body that evokes an immune response either alone or after forming a complex with a larger molecule. Antigen includes toxin, bacteria, foreign blood cells, and the cells of transplanted organs. Immunogenicity of blood group antigens. A, B, and D, and that is rho, are most immunogenic, which followed by Kell, Duffy, Kidd, etc. Antibodies. Antibodies are also called immunoglobulin Y-shaped molecules, are proteins manufactured by the body that help fight against foreign substance called antigen. Antibodies are of two types: naturally occurring or expected antibodies. If an ABO antigen is missing from an individual's red cell's membrane, then it is expected that the individual will produce an antibody to that antigen. They are called naturally occurring or expected antibodies. Example: anti-A, anti-B, anti-AB. Atypicals or unexpected antibodies. In all blood group systems, other than ABO, if the antigen is missing from the red cells, the individual is not expected to produce an antibody against it normally. When these antibodies are produced, they are termed unexpected or atypicals or allo antibodies. These antibodies are normally of IgG class. Example: anti-D, anti-C, anti-FYA, etc. Blood group genetics. The ABO gene locus is located on the chromosome 9 and RH gene is located on chromosome 1. H gene is located on 19 chromosome. It encodes for FUT1 enzyme, also known as fucosyl transferase, which mediate additions of fucose to a type 2 oligosaccharide chain, forming an antigen H, a precursor to each of the blood ABO blood group antigen. Blood compatibility test. Compatibility test is also called pre-transfusion test. It is always performed prior to blood transfusion in order to help prevent hemolytic transfusion reactions which may be caused by antibodies of the ABO blood group system or by antibodies to the other blood group antigen. Compatibility testing includes ABO and RH typing, antibody screens for unexpected antibodies, cross-match between donor red cells and recipient serum. ABO typing. Determinations of ABO blood groups is the most important pre-transfusion compatibility test. ABO typing is accomplished by testing patients' red cells with anti-A and anti-B antibodies, also known as forward typing. Testing patients' serums for anti-A and anti-B, which is also known as back or reverse typing. Antibody screening. The patient's serum is tested for the presence of clinically significant antibodies using an indirect antiglobulin method. The serum is tested against unpooled group O cells selected to possess the relevant blood group's antigens. Cross-match. The cross-match is routinely used as the final step of pre-transfusion compatibility testing. Cross-matching will detect incompatibilities between the donor and recipient that will not be evident on blood typing. There are two types of cross-match: major cross-match and minor cross-match. Major cross-match. The major cross-match involves testing the patient's serum with donor cells to determine whether the patient has an antibody which may cause a hemolytic transfusion reaction or decrease cell survival of donor cells. This is the most important cross-match. Minor cross-match. The minor cross-match involves testing the patient's cells with donor plasma to determine whether there is an antibody in the donor's plasma directed against an antigen on the patient's cells. Minor cross-matches are rarely performed. Hemolytic disease of the newborn baby. It is also known as hemolytic disease of the fetus and newborn, or HDFN, or HDN, or erythroblastosis fetalis. It occurs when fetal red blood cells, or RBC, which possess an antigen acquired from the father that the mother lacks, cross the placenta into the maternal circulation, where they stimulate antibody production. The antibodies return to the fetal circulation and result in RBC destruction. HDN was first described by a French midwife who delivered twins in 1609. One baby was swollen and died soon after birth. The other baby developed jaundice and died several days later. Types of HDN or hemolytic disease of the newborn baby. 1. Rhesus HDN, 2nd. ABO HDN, 3rd. Kale HDN, 4th. Kid HDN, etc. RHD HDN. RHD HDN is often called Rhesus disease and is the most important common form of severe HDN. It occurs when a mother with Rh negative blood becomes pregnant with Rh positive baby that inherited from Rh positive father. During late pregnancy and especially during the process of childbirth, the fetal red blood cells may escape into the mother's circulation. Once these cells having antigen D reach the mother's circulation, they are perceived as foreign and therefore anti-D antibodies is stimulated in mother's plasma. Rh sensitization normally is not a problem with the first pregnancy and so the baby is unaffected. Most problems occur in future pregnancies with another Rh positive fetus. During the second pregnancy, the passage of Rh positive fetal blood cells to maternal blood causes a severe immune response. Large number of immunoglobulin G or Ig antibodies are produced in the mother that cross the placenta, reach the fetal blood and destroys its RBC. Rh cause HDN is less common but more severe. This is called erythroblastosis fetalis during pregnancy. Once the baby is born, it is called HDN or hemolytic disease of newborn baby. ABO HDN. ABO HDN or hemolytic disease is arose when a mother with blood type O becomes pregnant with a fetus with different blood types A, B or AB. In type O mothers, the antibodies are predominantly immunoglobulin G classes and can cross the placenta. It thus can cause hemolysis of the fetal cells. This condition can occur on the first pregnancy and can occur without having been transfusion. With maternal blood types A and B, isoimmunization does not occur because the naturally occurring antibodies anti-A and anti-B are immunoglobulin M or IgM class, not IgG. Occurrence of ABO HDN is more common but less severe. ABO HDN does not become more severe after further pregnancies. Some common symptoms of HDN. After birth, symptoms in a baby include pale looking skin due to having few red blood cells, also known as anemia, yellow coloring of a baby's umbilical cord, skin, and the white of his or her eyes, also known as jaundice. Enlarged liver or spleen. Newborns with hydrophytalis may have severe swelling of the entire bodies due to fluid accumulations which can lead to heart failure. In conclusion, using the principle of immunohematology, we can successfully transfuse blood from a healthy donor to a recipient patient safely with great accuracy. Some of the tests which are done commonly before blood transfusion are blood group typing, Rh typing, antibody screening of a recipient's patient, and followed by cross-match. By virtue of immunohematology, we can diagnose many diseases such as hemolytic disease of newborn baby, AIDS, hepatitis, etc. And hence, suitable treatment can be done accordingly. Rh-HDN is the most severe hemolytic disease, which occurs when Rh negative mother carries Rh positive fetus. Through the highly successful use of prophylactic Rh-D immunoglobulin or ROGAM in Rh-D negative women, Rh-D alloimmunization and its effect have greatly diminished. As a result, abioincompatibility is now the single largest cause of HDN in the world.